Hello everyone, we are from Group 1. Today we discuss about gene cloning in plasmid. Do you know what is gene cloning? What is plasmid? And how the gene is cloned?
No!
Now, I will tell you all about what is gene cloning. Gene cloning deals with identification and isolation of desired gene and then joining the gene of interest into another organism. Multiple DNA cloned up into a single vector in a predefined order, orientations, and reading frame is known as multiple clone genes. Why gene cloning is performed? It is to study the clone DNA or produce the protein encoded by the genes.
Okay, now I will introduce you what is plasmid. Firstly, plasmid are circular pieces of DNA found naturally in bacteria. It can also carry genes, gene receptors, toxins, or other proteins. Plasmid are extrachromosomal DNA molecules that replicate separately from the chromosome of organism. It can also be engineered to be useful cloning vectors. The figures show plasmid may be used as vectors, which are small DNA vehicle that transfer foreign DNA to a host.
Now, for the last part, we will briefly talk on how the gene is cloned. It will involve in 4 steps. First is by identification and isolation of desired gene. Secondly, by insertion of desired gene into vector. Then, by transformation, and lastly followed with multiplication.
Now, I will explain more further about the steps in gene cloning. The first step in gene cloning is to isolate the DNA from the organisms that contains the desired gene. The second step is insertion of desired gene into vector. Insertion of gene into vector is hybridized by restriction enzyme. The next step is to incorporate the plasmids into bacterial host cells by transformation.
And last but not least, by demultiplication. Hosts will replicate together with plasmid and producing clone gene of interest in multiple copies. So by now, we hope you guys will understand more about gene cloning in plasmid. That's all from us. Bye!